All right, well, thanks so much, Lena. As Lena said, my name is Sarah Lamore. I'm a scientist in the Drug Safety and Metabolism Department at AstraZeneca. And today I'm going to give you an overview of how we are using impedance technology to predict cardiotoxicity. And specifically, I'm going to highlight a study that I have been working on to deconvolute and predict kinase inhibitor CV toxicity. So if we take a look at reasons for drug attrition and withdrawal over the past 30 years or so, toxicity and clinical safety issues have remained a major contributor to drug discontinuation. This chart is derived from a few separate reports, but it's really meant to illustrate the high proportion of drugs within that fraction that have failed due to cardiovascular toxicity. These CV safety issues have persisted despite the in vivo and ex vivo models that we have with excellent translation. So if we could add to the arsenal of preclinical safety screens by developing frontline in vitro assays that are capable of driving structure-activity relationships, this would enable chemists to design in safety. A key example of this is early hERG screening, which has dramatically reduced prolonged QT. However, there has not been as much success in early detection of functional CV risks, where there's a gap in frontline strategy for assessing beating function, such as contractility. Prime case in point is left ventricular dysfunction and heart failure that have been seen as a result of many of the tyrosine kinase inhibitors. And in many cases, this was not predicted with existing preclinical models. So how can we improve preclinical safety models to better predict cardiotoxicity? To give you an outline of my talk, as Lena said, first I'll give a brief overview of impedance technology. I'm sure there are many listeners who are users of the technology, but for those who are not as familiar, I'll give a brief overview. I'll then describe a validation study where we've used impedance assays for in vitro assessment of cardiotoxicity, and then go into the postdoc project that I had worked on aiming to deconvolute kinase inhibitor cardiotoxicity using induced pluripotent stem cell-derived cardiomyocytes and impedance. So a brief overview. Almost all impedance instruments use microtiter plates. The bottom of each well is coated with interdigitated gold electrodes, and when cells are seated and adhere to the bottom of the well, a very slow alternating current is applied, and the cell monolayer impedes the current. So this is very similar to resistance, but because we're applying alternating current and not direct current, it's referred to as impedance. So when the wells are empty, the impedance is essentially zero, and as cells are seated and attach and divide, the impedance becomes greater and greater. Now, the electric current can take one of three paths. It can go in between the cells, through the cells, or around the cells. And this offers a very sensitive detection of changes in cell morphology, cell-to-cell contact, and cell adhesion. So there are many advantages to impedance assays. It is very, very sensitive and can allow for detection of morphology changes down to about 1 nanometer. And to put things into perspective, the cell membrane is about 3 nanometers, and light microscopy has a resolution of about 250 nanometers. In addition to its sensitivity, you're monitoring whole cells in a label-free format, and therefore analysis is non-invasive and free of artifacts from exogenous labeling. And you're performing this in real time, so you can get continuous monitoring for response dynamics. So this really allows for assessment of a wide range of cell events and signaling cascades. The xCELLigence instrument captures data at a very high speed, every 12.9 milliseconds to be exact. So that means that you're getting close to 80 data points per second. So when cardiomyocytes beat spontaneously and synchronously in culture, as you see here, the rapid data collection of xCELLigence easily allows for detection of cardiomyocyte beating in real time. So this here is a movie showing a recording of 12 wells in real time as the cardiomyocytes beat, the change in morphology is detected as a change in impedance. Now, this here shows a close-up of a raw trace of impedance as a monolayer of cardiomyocytes beat. There are several beat parameters that we can take a look at, and beat rate and amplitude of the change in cell index as the cells beat are the two major parameters that we have focused on at AstraZeneca, and those are the two that I will be talking about today. So if we take a look at untreated cells on the top versus treated cells, we can see very clearly drug-induced changes, for instance here an increased beat rate and a decreased amplitude. We can get very quantitative pharmacology with very robust concentration response curves that span a wide potency range. For instance, an increase in beat rate in response to calcium channel activators or adrenergic agonists, or a decrease in beat rate in response to sodium channel blockers or muscarinic agonists. So can we utilize this technology to fill a gap that hasn't been addressed? And specifically, can we evaluate functional cardiotoxicity, perhaps looking at contractility? AstraZeneca's gold standard for in vitro preclinical safety assessment of cardiac contractility has been the dog IonOptix assay. This was validated a few years ago here at the company. It's an optical-based assay that measures sarcomere shortening in freshly isolated paced dog cardiomyocytes. And as I said, it's been validated using a set of 50 compounds with known in vivo effects on contractility. This is a very time-consuming and expensive assay. So we wanted to compare the impedance assay versus the dog sarcomere shortening assay. And you can see that there's very good correlation between impedance data, between the IC50s obtained with impedance data on the y-axis versus the IC50s that we obtained with dog sarcomere shortening. This has recently been published by Clay Scott in ToxSci. And the take-home message is that this can be used as a contractility correlate, and it corroborates very well with in vivo data. So these data and this validation study leads us to believe that the impedance assay using human stem cell-derived cardiomyocytes can help to fill the gap in preclinical screening for early detection of functional CV risks. Now, kinase inhibitors in particular have persisted as a problematic class of drugs, leading to adverse cardiac functional effects in the clinic. In vitro detection of these effects has been very challenging, and that's probably for several reasons. First, there are over 500 kinases in the human kinome, and most have a conserved ATP binding domain, leading to inherent promiscuity of kinase inhibitors. And this really creates a need for throughput to seek preferred chemistry. In contrast to hERG or other ion channels, kinases have pleiotropic cellular functions, and this creates a need for assays with integrated endpoints downstream of cardiomyocyte contraction cascade. Large panels of human kinase assays are now available, but this generates a need for human-based model to match these data. Now, the impedance assay seems well poised to address the demands on assays for kinase inhibitor cardiovascular toxicity. So to test this assay, I looked at compounds from kinase inhibitor programs that failed late in vivo. The first is from a microtubule affinity regulating kinase inhibitor program. Due to their role in phosphorylation of tau, MARK inhibitors are an attractive target for the treatment of Alzheimer's disease. Chk inhibitors, because of their role in DNA damage repair, are a logical and attractive target for anti-cancer therapy. However, compounds inhibiting these kinases have had unresolved functional cardiotoxicity that was discovered late in vivo or in clinical trials. So I used these compounds in a pilot study to see if impedance assays could deliver on 3 core needs. Could we detect cardiovascular effects earlier than in vivo or in the clinic? Could we start to understand the mechanism of action and determine if it was an on-target versus off-target effect? And if it was off-target, could we use this to develop an SAR-driving assay? And I'll show you data from both of these proof-of-concept experiments. So for the first example, looking at MARK inhibitors targeted for Alzheimer's disease, where toxicity was first detected in anesthetized dogs, the first question is, can we detect effects on cardiomyocyte beating in our assay? Yes, we can. There's a decrease in beat amplitude, and the IC50 correlates with the 10-fold change in enzyme affinity. These cells stop beating, and this is not due to cytotoxicity as measured by ATP and troponin release. So they're alive, they're just not beating. The alignment between beating cessation and enzyme inhibition suggests on target. But the second question is, can we determine if this is due to on or off target? So siRNA-based knockdown of the MARKs shows a decrease in beat amplitude if MARK2 or MARK4 are knocked down. And an even more striking effect is seen when all 4 MARKs are knocked down together. Now, the canonical role of MARKs is regulation of microtubule dynamics. So if we inhibit MARK, we expect to inhibit microtubule dynamics. So using pharmacological microtubule inhibitors, we also see an effect on beat amplitude. So both the genetic data and the pharmacological data suggest that these CV effects are at least in part due to an on-target class effect. So if we look at the second example of checkpoint kinase inhibitors targeted for anti-cancer therapy, however, 2 molecules, including an AstraZeneca compound, AZD7762, both had dose-limiting toxicities in the clinic, and neither one of these molecules progressed past phase 1. So can we see anything in the cardiomyocyte impedance assay? Both potently inhibit beat amplitude, and again, this is not due to cytotoxicity, so these cells are alive, they're just not beating. However, when you start to look at other Chk inhibitors, there's a very broad range of effects on beat amplitude. The IC50s span about 1,000-fold, and some molecules have no effect on beating. So is this effect target-related? Are these differences due to differences in enzyme affinity? In comparing the beat amplitude IC50s on the y-axis versus the kinase inhibition IC50s for both Chk1 or Chk2, we see that there is no correlation for either of these enzymes. So the pharmacology suggests the effect is due to an off-target mechanism. But what about knocking down the Chks? So when we knock down either Chk1 or Chk2 or the 2 together, we do not see any effect, suggesting that this is an off-target mechanism, perhaps due to inhibition of some other kinase or kinases. So using these compounds that failed in vivo, we were able to show that this assay could detect functional cardiotoxicity in vitro, determine if the effect was on target versus off target, and if the platform was compatible with an SAR-driving assay. So given the success of this proof of concept study, I'd like to describe an ongoing study where I'm using this technology to scan the kinome for functional cardiotoxicity and deconvolute kinase inhibitor-induced cardiotoxicity. So in thinking about this, there are several hypotheses or several factors that can contribute to kinase inhibitor cardiotoxicity. Given the inherent promiscuous nature of kinase inhibitors, Perhaps it is all about kinome-wide promiscuity, where the more kinases you hit, the more likely you are to have a problem, and that would be a very good predictor. Or maybe it's kinases within a particular group. So if we hit enough kinases within one specific group, maybe the tyrosine kinases, that's when we can expect to see a problem. Or maybe it's that it's a particular pathway, or it is a very relatively small number of kinases or multi-kinase profiles that are the most valuable predictors. So to go about testing these hypotheses, I have a large panel of kinase inhibitors where we know the number of kinases inhibited by each compound, and we know the binding affinity of each compound for about 385 kinases. This was published by Davis et al. In 2011. And so if we combine this kinome-wide data with the functional effects of these compounds as measured by impedance, can we identify culprit kinases or kinases critical for cardiomyocyte function, and ultimately develop a predictive model for kinase inhibitor cardiovascular toxicity? Now, I tested these compounds at a concentration of 3 micromolar to match the binding affinity data, and I measured effects after 2 hours. And if we ask the first question, is kinome-wide promiscuity a good predictor of functional toxicity? If we were to plot effects on beating versus a selectivity score, which is indicative of promiscuity, where the higher the F score, the more promiscuous the compound is, what would this data look like? And what we see is that there is a very weak correlation between functional effects and selectivity score or F score. But rather what we see is compounds, or rather selective compounds, that have an effect irrespective of promiscuity. Now, can we use this data to deconvolute kinase inhibitor cardiotoxicity? I'll come back to that in a minute. But first, if we look at intergroup promiscuity, one might expect that promiscuity within one particular kinase group, perhaps the tyrosine kinase group, correlates with cardiovascular toxicity, because many of the tyrosine kinase inhibitors do have functional CV side effects in the clinic. So in analyzing this data, we found that correlation of intergroup promiscuity correlating with functional effects as described by the Matthews correlation coefficient. The Matthews correlation coefficient, where 1 describes a perfect prediction and 0 describes a prediction no better than random. We found that promiscuity within 4 of the groups was correlated with effects on beating: the CAMK, the other, the STE, and the AGC groups. So there are 2 interesting things to me about this. The first is that it's not tyrosine kinases that one might expect. And the second is that 4 out of 10 groups are correlated, or intergroup promiscuity within 4 of the 10 groups is correlated with functional cardiotoxicity. So this might not be the best predictor. These 4 groups comprise about 50% of the kinome. So perhaps this is not conducive to prediction. So can we identify specific kinases that are correlated with cardiotoxicity? In order to answer this question, we use feature importance algorithms to identify univariate correlation between kinase inhibition and beating activity. And this, again, is described by the Matthews correlation coefficient. We identified 46 kinases that are correlated with functional cardiotoxicity and expressed in the heart and in induced pluripotent stem cell-derived cardiomyocytes. Now, this inhibition of 46 kinases, these 46 kinases are a very, very good start. But can we get it down to a smaller